Hello everyone and welcome to today's live broadcast, Disparities in Colorectal Cancer, presented by Dr. John Carethers, who is a John G. Searle Professor of Internal Medicine and Professor and Chair in the Department of Internal Medicine, Professor of Human Genetics at UMHS Internal Medicine in the University of Michigan. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located in the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of our presentation. Also, please notice that you are viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower left. If you have any trouble seeing or hearing this presentation, click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. This presentation is educational and thus offers continuing educational credits. Please click on the button at the bottom left-hand corner and follow the process for obtaining your credits. Without further ado, please welcome Dr. John Carethers. I will now turn the presentation over to him. Welcome, sir.
It's my pleasure to present some data and some thoughts on disparities in colorectal cancer and hopefully educate you on this very field. So as with most cancers, cancers develop in a genetically susceptible individual, meaning you have the genetic makeup that could possibly develop cancer. However, typically, unless it's a classic germline mutation in which puts you at extremely high risk, most of these put you at a low risk. However, if you have the right exposure that interacts with that genetically susceptible individual, you can develop a cancer. And that's what I would call the gene-environmental interaction. And here's some examples on this slide. You could take a genetically susceptible individual, expose them to human papillomavirus, and you could develop cervical or anal or head and neck cancer. Likewise, and not going through every example, you could have someone who has developed inflammatory bowel disease, so a level of inflammation, and ultimately develop colon cancer. Not everyone who's genetically susceptible develops a cancer, but the right interaction can make that interplay and then allow that cancer to develop. Even everyone who smokes doesn't get a lung cancer, although that's the leading cause of lung cancer, but not everyone who smokes gets a lung cancer. They may have other lung diseases, but certainly in a genetically susceptible individual, your chance of getting lung cancer are vastly higher than someone who doesn't smoke. In the colon, the process of developing a cancer usually starts at the polyp level. And on the top of this slide, you can see that there's a depiction of progression from a hyperproliferative state where you get a small bump on the inner line of the colon, and that could grow due to uncontrolled proliferation in a benign state until you get to the status of a larger polyp that you could see in these 6 examples here at colonoscopy. If that polyp continues to grow and proliferate, it can acquire other mutations within the cells of that polyp that can turn into more of a malignant state, or what we call cancer, and then that cancer can invade and spread. And on the bottom of the slide are 6 endoscopic examples. These are pictures taken at the time of colonoscopy, examples of what we see. For instance, in the upper left here, So this is a typical exophytic adenoma there. In the middle panel here, this is a little more complex. It's oblong, but this is still a benign lesion. And then on the upper right here, you'll see a very large adenoma with a very fat exophytic base, and this is still a benign. In the bottom left, here's another example with a long stalk with the adenoma at the very tip there. Now in this bottom middle panel, this is much more of a flat lesion, and this was biopsy to show that it had cancer. So this started as a polyp that might have been flat, but then turned into cancer. And then on the bottom right here is another example of a very, very flat, variegated, widespread lesion that turns out it was a cancer. On the next slide are a few more examples of this, even with high-resolution colonoscopy. In the upper left, a typical small polyp. This is probably the most common thing we see when we do colonoscopy and those appropriate for colonoscopy. In the middle panel, another little more complex adenoma. And then on the upper right here, here's a large example of a polyp, an adenomatous polyp that has an ulcer in the middle of it there that turns out that was where the cancer started. In the bottom left are a specific type of adenomas we call sessile serrated polyps. They tend to be on the right side of the colon, and with high-resolution colonoscopy, you can even see the pitting patterns on the surface of this serrated polyp. In the bottom middle, another example with under narrowband imaging. This is a special technique where you have only certain light shown, and you can see the vascular pattern of the polyp. And in the bottom right, here's one with a sessile serrated polyp, of which the yellow arrows show an area of invasive cancer. Sometimes these can be very subtle. But to suffice it to say that most colon cancers develop from the polyps. And the type of polyps that progress to cancer are typically adenomas or sessile serrated adenomas. And these are defined as these exophytic lesions that are already dysplastic under the microscope if the pathologist looked at them. And it's thought to progress from the normal to this polyp stage to a cancer stage over decades for most people. Now, on this next slide is an example of cancers. So I showed you some cancers in a polyp, but the two examples at the top of this slide are very large lesions that even partially obstruct the, partially or fully obstruct the entire lumen of the large intestine or colon. In the upper right, this covered 50% of the lumen, a very large cancer. And on the upper right, one that what we would call an annular lesion that's grown across the entire circumference of the colon. On the bottom right depicts this in a more graphic sense, and this is taken from the National Cancer Institute website, that shows early stage where the lesion is on the inside of the bowel, and if plucked off, is gone. But you get to stage 2 where there's invasion of the cancer invades the muscle layer that may require surgery to remove, and you get to stage, sorry, that was stage 1. Stage 2, in which it's gone through the muscle layer, and stage 3 has spread to some of the lymph nodes. And stage 4 means there's generally metastases that have spread beyond this organ. And so that's what we call staging, and that could determine your survival. The more advanced stage, the less likely you survive long term. Now, how common is colon cancer? In the United States, it's estimated that there will be over 135,000 people who develop colon cancer in 2017, and 50,000 will die from colorectal cancer. Now, I happen to be at the University of Michigan, and we have the largest stadium in North America, and that's Michigan Stadium, and that capacity of that stadium is over 107,000. Now, if I told you that everyone in that stadium would die this year, And then another third of the stadium, you'd probably be greatly appalled. But that's how many people die of colorectal cancer. It's a stadium, it's a 1.3 of these stadiums. And if I told you half, that will get colon cancer, I'm sorry, but half the stadium will die from colon cancer. That, you can get a sense on how many people that is when you put it in that context. Now, the best way to take care of cancer is not to get it at all. And for colorectal cancer, that is generally done by screening. Now, what is screening? Screening is a way to try to find the precursor to cancers and prevent the cancer from forming. But it must meet some general guidelines. That is, whatever test that you use for screening should be sensitive and be able to detect what you are testing. So in other words, you're able to find it if the test is correct to test for that. It should be specific. In other words, does not detect the cancer when negative. And it should be a condition that's broad enough, common enough, and represented in the population. If you're the rare bird that it's 1 in 200 million persons, no one's going to screen 200 million people to find that one person. But if in colon cancer the numbers are large, screening can prevent that. It should be cost effective. If you spend $10 million per person to do screening, that's not going to be cost effective for society. Also, the test should do little or no harm. In other words, whatever test you pick for screening shouldn't cause more morbidity than the thing you're trying to prevent. And it should reduce mortality. In other words, the test should prevent you from developing the cancer, and thus you would have a normal lifespan. And so this is absolutely true for many types of screening, including colon cancer, colorectal cancer. Here is a slide that just shows 4 common cancers that we screen for: breast in women, cervical cancer in women, prostate cancer in men, and colorectal cancer. Now, if you look at the numbers for the incidence in the United States in 2017, you see that colorectal cancer is not the most common cancer. It's actually behind lung, which is not on this slide, but of the gender-based cancers, breast and prostate are more common. But if you look at the deaths of these commonly screened for cancers, it is the most common cause of death, more so than breast and prostate. And if you look at the types of screening, the types of methods to screen for these cancers, breast cancer is largely mammography. And in the literature and in the media recently, there's been some controversy on the age of screening because of the ability for the sensitivity and specificity of mammography. But it's still a decent screening method when used appropriately and at the appropriate ages. Cervical cancer is generally screened by Pap smears. So there's one test for both of these. Prostate cancer screening had been PSAs, but that's intercontroversy, and its sensitivity and specificity is questioned. And so there are other methods being detected for screening. That in combination of a rectal exam is generally what has been used in the past. But look at colorectal cancer. It's so much a common cause of death that we've invented 7 or 8 tests for that, each with its different sensitivity and specificity. And more recently, based on the July 2017 guidelines published in Gastroenterology, they have broken down the types of tests that would be first tier for most patients in the United States, and that would be the fecal immunohistochemical test and colonoscopy for most patients. And for some reason, that would not be adequate, or there's patient preference. The second tier test might be appropriate, which would include CT colonography, a fecal DNA test that's usually in combination with a fecal immunohistochemical test, and or flexible sigmoidoscopy. And then, and rarely you would go to a third tier test like capsule colonoscopy. And I'll touch base on each one of these and their sensitivities shortly. And the tests that are now relatively obscure or obsolete are fecal occult blood tests, which have now been largely replaced by the FIT test, and barium enemas, which its sensitivity has been relatively poor, and most people do not like to take the substance of an enema, which may not actually look at the entire colon anyway. So times have changed. Now, just going over these types of tests, the strategies for screening in the United States is the biggest one is probably colonoscopy. And generally, if you have a colonoscopy that shows no polyps, you would have intervals, and you don't have a strong family history, you would have an interval of 10 years if that colonoscopy was negative for polyps. It is very sensitive for significant lesions. That's large polyps and cancers. And its sensitivity, depending on which paper you read, ranges from 85 to 97%. It is the test that is also therapeutic, so it gets a good look based on the operator doing the test, but it also can remove those lesions. And in fact, there's lots of evidence by removing polyps that you reduce greatly your risk for getting colorectal cancer. However, because it's uncomfortable, there is usually some conscious sedation or some form of anesthesia given. It requires a prep, and there is a modest risk for perforation of the colon using an instrument. But it's the only modality for surveillance for high-risk patients. In other words, if you've had polyps before, or had a cancer before, or has had one of these strong family histories, it's colonoscopy that is the only test that should be recommended for you in those situations. For routine screening, it is one of the tests. The other one that has nearly the same sensitivity for significant lesions is CT colonography. This is another test that uses non-invasive X-rays to render the colon in 3D, and you can get a virtual fly-through of the colon. However, it does require prep because you got to remove the feces from the colon. It discounts small polyps, and it's diagnostic only. So if something is found at CT colonography, it requires a colonoscopy. Now here's some examples of other tests. Some of these are less invasive or non-invasive. A flexible sigmoidoscopy, which is a shortened version of the colonoscopy, does not require the same prep as a colonoscopy. It generally requires enemas. Its detection of significant lesions is on the order of 70 to 85%, and only through the left colon or sigmoid colon. It does not reach the transverse or the right colon, so it does not detect lesions there. So it's 0% in those areas. And if you find something on that, generally we do a colonoscopy, so you have to undergo a second test. If you combine tests like flexible sigmoidoscopy and a FIT or FOBT test, its sensitivity is not that much different from a sigmoidoscopy, with the idea that the other non-sigmoidoscopy tests would detect lesions on the right side of the colon where the sigmoidoscopy couldn't reach. But if any of those tests positive, it still requires a colonoscopy. A barium enema, I mentioned, is pretty much obsolete. Its sensitivity was not very good, on the order of 33–40%, and I'm probably generous on this slide. But if it's positive, it requires a colonoscopy. And the totally non-invasive tests, like fecal immunochemical test, its sensitivity is 25 to 71%. If it's positive, it requires a colonoscopy. FOBT, which has been largely supplanted by FIT, has a poor sensitivity. It's inexpensive though. And the more recent stool fecal DNA tests are more expensive. Their sensitivity is pretty good and maybe even better than a FIT, but its cost is what's relatively prohibitive. And of course, if that's positive, it requires a colonoscopy anyway. There's a couple other tests which are not generally recommended, but I'm going to mention them. Capsule colonoscopy is not FDA approved for colon cancer screening. It's only approved if the imaging of the right colon is not complete at colonoscopy. But its sensitivity for adenomas is fairly decent at 88%, but it's poor for sessile lesions or lesions, particularly on the left side of the colon. There's also a blood test called the Septinine assay that was developed but its sensitivity is on the order of 48%, so very, very poor. And that's only for cancers. It's 0% for adenomas. And that's, you really want to detect the benign precursors of the cancer in order to remove the risk completely. And there's some studies that suggest it may be sensitive for late-stage cancers, not even early-stage cancers, in which you want to find at the worst-case scenario during the screening method. So these are just several screening strategies that I present here. Now, you know, colorectal cancer, I mentioned 135,000 people get it, 50,000 people die from it annually. Your outcome is related to the stage. I mentioned that earlier. Thus, if you can detect either early stage tumors or the adenomas, because these cancers generally begin as these adenomatous polyps, and removal of adenomas should reduce your incidence as well as your mortality from colorectal cancer. And that's certainly the case. In the United States, colonoscopy has become the preferred method for most people, although there's some patients that don't like the minimally invasive strategy for this. And this picture just depicts colonoscope and how it traverses through the colon with the use of an operator or an endoscopist. And in the bottom photo there, it shows the act of polypectomy, in which we use a snare to either cold or hot snare, depends on if there's electrical cautery put through it, can remove those polyps. And keep in mind, colonoscopy is diagnostic and therapeutic. It's can be used for screening and surveillance, meaning your average risk for sporadic or familial, and polypectomy, it's the preferred method for polypectomy, which is the most common procedure. It's more expensive than obviously a FIT test or FOBT test, but because it is diagnostic and therapeutic, it is still relatively cost effective. Now, one of the things about using this as a screening modality is, does it reduce mortality? The answer is yes. There have been, this was really the definitive paper. This is data from Ann Zauber and a national Polyps study, which is a large cohort study that was done out of New York. And this table taken from the New England Journal of Medicine article shows that if you look at all comers for fault time and looking at the observed deaths compared to expected deaths for those who got colonoscopy compared to those who were in the general population, that the reduction of deaths is on the order of 53%. And that didn't change very much whether you looked at someone who had a colonoscopy less than 10 years or greater than equal to 10 years. It was in the 51 to 56 percentile, and those were statistically significant. So the answer is yes, colonoscopy can and does reduce your chance of dying from colorectal cancer, and it's largely through the removal of those adenomatous polyps. The United States Preventive Services Task Force just about a year ago did a complete review of many of the screening modalities used to screen for colorectal cancer or colorectal adenomas. And this table, which I apologize for being somewhat difficult to read, just basically goes over the frequency for all these tests and the evidence of efficacy and some other considerations. So in looking at the stool-based test, guaiac fecal cut blood tests, where you take a sample of the stool, pass it home, and put those on the cards and mail those in, does not require bowel preparation. It's cheap, but its sensitivity is on the low side. The fecal immunochemical test is now in the first tier for the multi-society recommendations. It's relatively inexpensive. Again, does not require bowel preparation or anesthesia. And you can do that at home and mail that in. And it's still relatively expensive, but its sensitivity is better than the FOBT, and it largely supplants the FOBT. The stool DNA test, that's the third one, FIT DNA, because it's usually combined with the FIT. The United States Preventive Services Task Force recommends that there's insufficient evidence about the longitudinal follow-up because it hasn't been longitudinally tested, but they also show evidence that it's expensive, and other people have shown that it may become cost-effective if the cost for particularly the DNA stool piece of that comes down. The direct visualization tests shown below, I touched base on these already: colonoscopy, CT colonography, flexible sigmoidoscopy, and combined flexible sigmoidoscopy with FIT, and generally are, for the most part, good screening strategies. The colonoscopy is a little more expensive, requires anesthesia and prep. CT colonography requires prep, but it's only diagnostic. The flexible sigmoidoscopy only covers the left side of the colon. And even with a combination of flexible sigmoidoscopy and FIT, that test is probably about a 75% sensitive as compared to colonoscopy alone. In their screening recommendations, they suggest that the target population be between age 50 and 75. The risk assessment, this is for the vast majority of average risk adults. The most important risk factor for developing colorectal cancer is age, but other things such as family history and being of African American race should be in considerations. I'll come back to that a little bit later. And they also said that there's no head-to-head studies on some of these tests, so they're not sure which one's the most effective one, although in the United States, colonoscopy has become the adopted gold standard. And other considerations such as health and prior screening status should be taken into consideration for those who have gotten much older, such as over 85 or 86. In fact, they recommend that those age 6 and older should never be screened anymore. And those between 75 and 85, that the net benefit for screening should be weighed in consideration. So really the target audience is between 50 and 75. And the average age of colorectal cancer in the United States is about 68. And that timeline from developing normal to polyp to cancer, the normal to polyp might take 40 or 50 years, so you get your first polyps, let's say, by the time you're age 50, and then it takes another 10 to 20 years for you to get cancer, and thus the average age is around 68. So this is not something that develops overnight. So what are the risk factors for developing colorectal cancer? Things that are non-modifiable and mentioned in the U.S. Preventive Services Task Force recommendations are age. The older you are, the more likely you're going to get polyps or cancer, and your family history. You have first and or second degree relatives that have had colorectal cancer or polyps. Your risk of getting colorectal cancer or polyps are elevated, and I'll show you that on an upcoming slide. The other thing that's non-modifiable is your race, and I'm going to show you some data that race, and in particularly in the United States, African American race, and in some cases Native Americans, have higher risk for developing these polyps, these adenomatous polyps, as well as higher risk of developing and dying from colorectal cancer. Now, theoretically, there's a lot of modifiable risks. Your diet, your socio-economic status, although that is harder to modify, your utilization for screening, healthcare access or insurability, your education, the use of tobacco and smoking. And I show you on the right-hand side of this slide that even though most people think of lung and laryngeal cancer as caused by smoking, the 10% of the risk of colon cancer is from tobacco usage. And I show that with that red arrow there. Other things such as non-steroidal anti-inflammatory drugs, which can lower your risk, as well as hormone replacement therapy, particularly in women, can lower your risk for cancer, and that has variable usage between even races and socioeconomic status. So there's potentially modifiable, but some of these are societal modifiable rather than non-modifiable, like your age, family history, or race. That's something that you can never change. So in thinking about age, there is approximately 5% prevalence of all Americans for adenomas that are greater than 9 millimeter by the time we're 50 years old. If you look at this curve shown on this slide, showing somewhere around the age of 50 years, about 25% of us who are that age or older will have an adenomatous polyp, this precursor lesion. And as you can see over time, as we age, the chances of getting by the time we're 75 is about 50%. So we develop adenomas, 25% of us will have them by age 50 already. Now, what happens with cancer? Now, cancer is a consequence of some of those adenomas, and we start screening. Recommendations are age 50, shown where I just put this arrow. But if you draw a line here at age 50, 95% of all colorectal cancers are after the age 50, with the average age of 60, as I mentioned, and only 5% of cancers are below the age 50. So you can see this curve goes dramatically up just around age 50, and this is why that age is picked for screening the general population. Now, I mentioned 5% of cancers are below the age 50. That is largely, could be sporadic, but that can also include familial high risk, where your genetic predisposition is extremely high for developed cancers at a young age. Now, so that's age. So your risk of developing adenoma and cancer go up. What about your family history? Well, this table I put together shows what your risk changes. So if you look at the averages, if there's no average risk of an American, no family history, it's about 1 in 18 to 1 in 20. So let's just say 5% of you developing a colorectal cancer somewhere between the age of 0 and 75 years old. So about 1 in 20. Your risk approximately doubles if you have a first-degree relative, that's mom, dad, brother, or sister, with either a cancer or an adenoma, because we know the adenoma is the precursor to cancer. So if you have a family relative, a first-degree relative, you've doubled your lifetime risk. If you have 2 second-degree relatives, so 2 people in the family, but maybe not mom, dad, brother, sister, but uncle, aunt, those type, any age with colorectal cancer, you've approximately tripled your risk. So family history is very important here. If you have a first-degree relative who has an adenoma, the precursor lesion, that's less than 60, you've tripled your risk. And over the general population. If you have someone who's young, relatively young, less than 45 years old with colorectal cancer, you've 6-fold increased your risk because someone has it at a very young age. Now, if you have one of these genetic syndromes in which the genetics dictate and drive the pathogenesis of cancer, such as Lynch syndrome or familial adenomatous polyposis, your risks of getting colon cancer are extremely high because of the genetics that you inherited. So 80% for Lynch syndrome, and 100% of people with FAP will get a colon cancer if their colon is not removed. So family history is very important for determining that. Generally, the guidelines up until July of 2017 basically said men and women, so the split of cancers between men and women are fairly equal, 71,000 for men, 64,000 for women, shown on the left part of this slide, who are asymptomatic and who are 50 years or older, and they have a negative family history, would get average risk screening, in which the new recommendations recommend one of the tier 1 tests, that's FIT or colonoscopy. If you have a positive family history and you have either first degree or 2 or more second degree relatives, then you change that age from 50 down to 40 years because of that family history. So we would start routine screening for those who have a family history at age 40. So the determinants are typically your age and your family history, okay? And there was no consideration of race until the recent guidelines just published in July of 2017. By the way, if you do have a family history, because it's unknown whether you might have a genetic syndrome or not, colonoscopy is the preferred test for that. Now, so I mentioned up until July 2017, race was not included in those guidelines. Now, this is data from the NCI, the National Cancer Institute, that looks at all races and then breaks up into the 5 main races in the United States: whites, blacks, Asians, American and Alaska Natives, and Hispanics. And if you look at the incidence, whether male or female, or the deaths from colorectal cancer, whether male or female, the highest is in the African American population by far. So looking at male, the all races is about 46.9 per 100,000 in the U.S. population. Whites are about that average, and black males 12 points higher. Asians have lower rates, and Alaskan American Natives are elevated above whites, but not to the level of blacks. Hispanics are even lower than the average. Same is true for females. And if you look at deaths, again, African American males and females, comparing to those races, other races, have the highest death rates for colorectal cancer. I do also want to point out, if you look at people aged 20 to 44, remember I said that there's about 5% of people get colon cancer below the age 50. Even those rates are still highest in particularly the black population in the United States. If you look at staging, so to what extent do people present with their colon cancer? Shown here on the left, so all races are in red, whites are in the green bars, and black patients are in the blue bars. So shown by the red arrows above that, so black patients for localized have slightly less localized disease by 1 percentage point, much lower regional disease, but much higher distant disease or metastatic disease. And these are standardized for age. This only difference here is race. If you look at the survivability, so again, red is all races, whites are green, and black patients are in blue. If you have localized disease, your survival rate, if you're black, is 4 points lower than if you were white, even with localized disease. If you look at regional disease, which is still curable by surgery, that was a 7-point lower survivability with someone with regional disease. And if you look at distant disease, which kills, shortens the survival for most patients, it was still even worse for black patients. And if you look at all stages, there's essentially an 8-point lower difference in survivability if you even come in with the same stage as other patients. So that's clearly, and this has been consistent data for the past 15, 20 years put out by the National Cancer Institute. If you look at the trends over time on this next slide, white patients are shown in blue and black patients are shown in the red bar. And this shows the rate per 100,000 over the span of 20 to 30 years. The black line shows goes up if there's a larger disparity between blacks and whites. So if you see that both blacks and whites rates for colorectal cancer have declined over the last 2 or 3 decades, but the disparity that they're declining at a slightly different slope, such that the disparity, shown by the black line, has gotten slightly larger, but it's pretty much leveling off, at least over the last maybe 10 years. If you look at women, again, both blacks and whites have declined, and the gap has narrowed such that the the disparity has stable to narrowed over time. Most people believe that this has to do with screening, but only time will tell that. So why are there racial differences? I just showed you on the previous slide. Some would argue, and it's probably multifactorial, but some would argue that socioeconomic status, insurance, and education limit the ability to have access to, say, screening. Blacks make up about 13–14% of the U.S. population, but 24% of the U.S. poor population, compared to 8% for whites. Access to medical care is different, and depends on if you're inner city, if you have insurance. Although the Affordable Care Act, which was put in place in 2014, theoretically could modify that, and that time will tell if that type of insurability is going to be continued or not. What about the use of colonoscopy? I mentioned colonoscopy is kind of moved to the gold standard in the United States, and you remove the polyps. There's some evidence that there is a right-sided excess of polyps in the African American population. And if, let's say, if the African American population was largely getting flexible sigmoidoscopies, they would miss out on that compared to other populations. Other things that are theoretically modifiable, but diets might be different amongst races, and depending on where you live in inner city or the food islands in which you're eating things that are not good for the local colonic environment, such as a high-fat, red meat, low-fiber type diet. The use of physical activity, the use of tobacco, the less use of hormone replacement therapy or NSAIDs, or even classic differences in biology or tumor metastases, including mistrust of the health system based on historical other experiments that have been done. Might explain some of these. It's largely probably multifactorial, but I'll explore a few of these things in the next slides. If you look at risk factors, and this is also taken from SEER data, that if you compare blacks versus whites on obesity, if you look at all black patients tend to be more obese, 48% compared to 36%. And that seems to be more dramatic in women, as shown in this data taken from the National Cancer Institute. Overweight, maybe not much different, but that's slightly less than obesity. What about physical activity? So no leisure time physical activity. I mean, are you more sedentary? And that seems to be higher amongst all blacks compared to whites, at least done in this survey. What about recommendations for aerobic activity? So this goes with the physical activity. Again, the numbers for blacks seem to be they need lower aerobic activity, meaning more sedentary lifestyle, which is more associated with developing cancers. And what about the use of tobacco? If you look at all blacks versus all whites, not much different, but black men had a couple percentage points higher for use of tobacco, which could contribute to the development of cancer disproportionately if that population uses more tobacco. What about screening usage? In other words, if screening usage is less, you're more likely to see cancer because you're not screening for them. And in this survey, also taken from the National Cancer Institute, looking at fecal occult blood testing, any form of endoscopy, which could be flexible sigmoidoscopy and or colonoscopy, or the combination of two. And the survey, which I think was done in 2014, was not dramatically different between blacks and whites at this stage. And this is actually good news, because in the past, their survey data showed that there was a disparity. This means more blacks are being screened and are comparable to whites. So based on this data, it might not be that just screening utilization itself is a driver for that disparity. The type of screening could make a difference, though, but based on this survey, there appears to be not much difference. What about the distribution of colorectal cancer? Well, a number of surveys and studies have shown that blacks tend to have more lesions on the right side of colon, or proximal colon, than whites. Data from the NCI through Searle in one study, this is a couple of decades old, but showed a 10% increase in the number of proximal significant lesions in blacks compared to whites. If you look at another study that came a decade later, blacks compared to whites, where whites were the reference, so there'd be 1.0. Asians had less on the right side with less likely, as well as Latinos. But blacks, they crossed that 1, and they were about 7% more likely in this study, compared to about 10% in the older study, of having a right-sided lesion, which are much more difficult to get to and may not be seen with certain screening tests. Now, if you look at this in how the polyps are distributed in another study, this is a study that looked at real-world 67 adult GI practices where they had over 85,000 participants, including blacks and whites. The two things to show here: blacks had 7.7% of significant polyps that were greater than 9 millimeters in size, the ones that are more likely to turn to cancer, compared to 6.2% for whites. So they had more advanced lesions, if you will. And if you look at black women versus white women in the circle, the odds ratio was 1.62 of a black woman having more likely to have an advanced polyp, and 1.16 for black men to have advanced polyp compared to white men. Now, I know this is a complex slide, but I'll just point to where the arrows. This is a study done on over 320,000 average risk adults, looking at 84 adult practices, looking at finding tumor polyps that were 9 millimeters or greater, and comparing blacks and Hispanics compared to whites. So if you look at either females or males, okay, and I've highlighted with the red arrows, almost every 5 years of age after the age 50, the red arrows mean the numbers were higher for black patients than whites. For females, essentially every 5 years was higher in the black, greater than a ratio of 1, except for age 70, 74 for some reason. And if you look at males, the younger ones did not cross the 1.0 threshold, but every other 5 years showed that blacks had much or were much more likely to show 9 millimeter polyps or greater, which are the higher risk polyps. And so this suggests, again, for standardized for age, that for some reason black patients tend to have more advanced polyps than white patients at the same age. It's not clear why. If you look at the distribution of polyps, the proximal or right colon versus the left colon in black patients, so a number of 50 for whites, it's evenly distributed, about 50% proximal, 50% distal. But look at the difference for blacks. 53% were proximal compared to 46% or 47% distal. And if you look at the other races like Hispanic, they had less on the right side. So this seems to be the pattern in many studies, that there were more advanced polyps in blacks and tend to be more of a proximal distribution for some of the polyps. This is a different study from the Kaiser Permanente, again looking at, I think on the range of 60,000 patients in this study. Again, a number of greater than 1 would mean, because white was the reference, would mean that there were more high-risk adenomas. And in this case, they broke down the races a little bit more granular. Blacks were 15% higher than whites, as shown in the other study. Asians were pretty close to whites, But here, Native Americans had the highest risk, even higher than blacks in this particular study. The numbers, if I recall, were small in terms of number of Native Americans, but this is significant. And the same thing occurred for women with similar larger numbers than whites, and as well as for men if you break it down by gender. So what this means is that is similar to the previous studies I showed you, that blacks tend to have more high-risk adenomas, and in this particular study, so do Native Americans. That Kaiser-Permanente study also looked at the right-sided distribution, or the distribution of polyps, and similar to the Lieberman study, there were, I know this is hard to read, but there were about 26% more polyps compared to whites on blacks, and I believe 55% for Native Americans for any proximal adenoma. And when you look at distal adenoma, Native Americans, blacks were 1.09, so 9% higher, and 40% higher for Native Americans. So there was a proximal distribution, but they also grew, because they grew more advanced polyps, they also grew on the left side as well. So, so what about developing the polyps on the right side, the adenomas on the right side? There seems to be increased right side prevalence. I just showed you some studies. Keep in mind, colonoscopy is not as effective in reducing mortality with right side lesions. Even though it has a complete visualization of the colon, there's been a number of studies showing that the sensitivity for reducing mortality on the right side is lower than on the left side. And it may have to do with flatter polyps, much more difficult to see polyps on the right side, and it's a further distance from the entry of the colonoscope. The combination of having right-sided polyps and difficult to see, even with the gold standard test of colonoscopy, could even amplify the magnitude of disparity with any condition which has excess right-sided cancers. So you tend to have more right-sided cancers, and you've got a test that doesn't detect it as well, that disparity will grow. There's also, I mentioned sessile-serrated polyps or adenomas early on. These are, there's very little race data on these. These tend to be more likely on the proximal. So one question is, are these excess proximal polyps, are they sessile-serrated adenomas, which have a higher risk of turning to cancer? They show microsatellite instability, MSI, but there's very little data on this. And the one study that had enough patients, it was 0.2% in Caucasians and 0.3% in African Americans, and there was no statistical difference. So sessile-serrated adenomas is probably not the game changer. So it's unclear why and what type of lesions are more on the proximal side in African Americans and possibly Native Americans compared to Caucasians. So the thought process is that some of these cancers, why are they more advanced? There's now data from several papers that suggest that the amount of cancers below the age 50 is much higher in African Americans. So remember I said earlier, screening starts at age 50, of which about 5% of the cancers are below age 50 and 95% above age 50. Well, if you map this for African Americans, it turns out that there's double that amount, so 10.6% of cancers in African Americans or blacks are below the age 50. If you started screening at age 45 for African Americans, you equate that 5% to the same curve. So if you started screening at 45 for African Americans, it would equate to the same number of cancers below age 50 if you start at age 50 for white patients. So overall, African Americans tend to present 0 to 8 years younger. It implies the adenomas were initiated at an earlier stage, so that's an implication, but there's no scientific evidence for that yet. And that means there's less time from age 50 to the average age of colorectal cancer, shortening the window for screening intervention. So there's been a proposal that will come up in a slide as I get near closing out that recommends moving the age of screening for African Americans down to 45. What about family history? Remember I said the screening algorithm, if you have first and second degree relatives, that you would start screening at age 40. Well, it turns out there's data, largely from the social medical media, excuse me, not social media, social medical literature, that African American and Hispanics with family history of colorectal cancer are least likely of all races to get screened. And African Americans are less likely than most races to know or transmit knowledge of family history of colorectal cancer or polyps. So it might be the dirty secret in family or they're strange or whatever, but that information is critical because that determines whether you are average risk and you screen at 50, or if you have family members, first degree and some second degree relatives, that would put you at age 40. So if you're not transmitting that data, you're going to get screened at 50 based on recommendations, unless you transmit that you have family members that present at a younger age, or have cancer or adenomatous in the family. Now there's another wrinkle to this too, because there are different pathways for colorectal cancer to develop. One is called the microsatellite instability pathway. Another one is microsatellite stable. It turns out that if you have the microsatellite instability pathway, which is about 15% of all colorectal cancers, you actually have better survival compared to those who are microsatellite stable. Those cancers do appear to be on right colon, so that could match African Americans. The mechanism tends to be methylation of one of the genes that cause microsatellite instability, called MOH1. But unfortunately, data suggests that this mechanism is less common amongst African Americans. So there's more right-sided lesions, but the MSI is not what's found there. And in fact, if you look at those that are microsatellite stable and particularly manifest another biomarker called EMAS, the survival is poor, and this is the one that tends to be more common amongst African Americans, despite it not necessarily being right-sided lesions. So it's still unclear why there are more right-sided lesions. They tend not to be this better surviving MSI, but most of the lesions that contain this EMAS, which causes poor survival, are more common amongst African Americans. Now, this is important because now there's therapies that can affect your outcome. So it turns out if you have the MSI high phenotype, which I just told you that it was less common in African Americans, it turns out there's a drug, an immunologic drug, that can further improve survival in those that have MSI high tumors. So if you don't have an MSI tumor, this drug doesn't work very well. So if you're in a population such as African Americans that don't get these types of tumors, this drug won't work very well. So this has implications on how patients are treated. Likewise, there's implications on other treatment. One of the mainstays of treatment for advanced colorectal cancer is a drug called 5-fluorouracil, or 5-FU. There's a number of studies that show, including studies we've done, that if you have an MSI high tumor, which is less common in African Americans, there is essentially no benefit of chemotherapy. And in fact, there's some studies that suggest if you give 5-FU in those of MSI high tumor, you shorten survival in some of these patients. Now, if African Americans have less MSI high, 5-FU would theoretically be better because there's less MSI high, more microsatellite stable, and they should improve. But yet, that still has not been demonstrated. In fact, the outcome is still poor. So this does not answer one of the questions of why they present present in a more metastatic state. but it does suggest that the biology of the tumor can dictate how these drugs could work. So overall, I want to say that colorectal cancers generally begin at polyps, but they can progress. Screening typically begins at age 50 unless there's a strong family history in which the age drops to 40 for those particular patients. Prior to July 2017, race had not been considered in the guidelines. But if you look at African Americans in particular, and to some degree Native Americans, there's a higher mortality, early age of onset for colorectal cancer. You look at the biomarkers of the MSI high, which is a good outcome. They have half the rate, African Americans have half the rate, but double the rate of the bad biomarker EMAS compared with whites. It has implications for the use of 5-FU and immune drugs. And there's also a higher number of right-sided polyps and cancers, but it's not clear on the mechanism or even the histologic formation of these tumors. And that's an area ripe for study. If you look at the cost effectiveness, and I know this is old data, but the principle is still the same, amongst racial groups, because African Americans present at a younger age and more advanced, they are the group that is the most cost effective for screening. Whether you look at a combination of fecal occult blood tests and sigmoidoscopy with a 5-year dwell, dwell time, or colonoscopy every 10 years with a 5 or 10 year dwell time, meaning how long it takes to develop those polyps or cancers. The African Americans or blacks are the lowest in terms of costs because they tend to present younger and more advanced compared to other races. So they're very cost effective. So cost is not really an issue here. If you look at principles for screening, if you have a high-risk condition, I mentioned FAP and Lynch syndrome, I can even mention inflammatory bowel disease or IBD. Most gastroenterologists and others are aware of that risk, and they will screen them or survey them appropriately. But the risk of African Americans is less likely to be known, and thus it's one of the reasons I'm giving this webinar, less likely known amongst gastroenterologists and other physicians because the data is not necessarily front and center there. But if you look at any high-risk individual, whether it's FAP, Lynch syndrome, or whatever, you would think, as long as it met those screening parameters of sensitivity, specificity, cost-effectiveness, etc., the screening would begin before the age of significant increases in cancer incidence for that particular group, whether it's IBD or whatever. And screening should be adjusted by the observed biology or epidemiology of the risk group. So for Lynch syndrome, we start screening them at very young ages, 25, doing colonoscopies, not every 10 years, but every other year. And by the time they're 40, they get it every year. And it's because we know the biology of Lynch syndrome. If you look at recommendations based on race up until July, the American College of Gastroenterology did recommend age 45 years. However, they weren't the body that most insurances look at to pay for that. The American College of Physicians, or ACP, even went lower and recommended screening start age 40 years for African Americans. And that's just to match, quote unquote, having a family history. The Institute for Clinical Systems Improvement recommended 45 years, like the American College of Gastroenterology. But the two shown in green, the Multi-Society Task Force and the U.S. Preventive Services Task Force, are the two agencies that really most insurances pay attention for and really change policy. Up until July, they both had recommended standard 15 years for African Americans, despite some of the data I showed you. Now, how could you reduce the disparities that I mentioned in African Americans? There are a lot of things that could be thought of. Patient education, if there's barriers to getting screened. Physician education. I mentioned not every, even gastroenterologists, know some of the issues that are specific for certain races. Patient navigation, that's a hot topic in which patients are offered screening, but because of life issues and other things, getting them to the screening site and getting the screening done is much more facilitated with patient navigation. And one of the pioneer areas is in the city of New York, where they're getting screening rates, even amongst minorities, to 80 percentile of utilization. But increasing screening age by any method, age 50, would increase the screening rates amongst African Americans. And we know that most colorectal cancers still occur after age 50. It might be the modality could be different, or a big difference amongst different races. Modifying the age for screening, as I suggested, some of the major societies recommended 45 or 40, would reduce the burden of early onset disease and raises awareness of that in that particular group, but does have the disadvantage of increasing confusion in guidelines. Again, most colorectal cancers develop after the age 50. However, in the published July recommendations by the U.S. Multi-Society Task Force, for the first time ever, they did include race to be considered in these national implementing guidelines. And they stated, and I quote, "Screening should begin at age 50 years in average risk persons, except in African Americans, in whom limited evidence supports screening to begin at age 45 years." So this is something that just happened in the past 2 months, we'll see if insurers will pay for it at age 45 versus 50 for screening. I will stop there, say thank you, and I'm open to taking questions right now.
Thank you so much, Dr. Carethers, for that informative presentation. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window, type in those questions into the box that appear on the screen, and click on the send button. We'll answer as many as we can before the end. So let's get started. We have a wonderful audience today. Our first question is, what might be the best strategy to lessen the disparity between races?
Question. I did mention everything from patient education, physician education, increasing screening after age 50 or lowering the age as examples. Probably it'll be some combination of those approaches. There's no question that patient and or physician education about some of the disparities, and I didn't even talk fully about Native Americans and their increase in a full manner, but I think education amongst all races for getting screening age 50 is beneficial, no matter what race. But to lower the disparity, that means targeting those populations. Lowering the screening age, as mentioned by the new guidelines, will probably help some and put some attention on that. But it will probably not reduce everything, because not only are people developing, particularly in African Americans, presented at a younger age, but the burden of disease is still high even over age 50. So I think you have to do a combination of those things, making sure your screening utilization approaches 80, 85, 90% of the population like they've done in New York with patient navigation, and perhaps lowering the screening age to put everyone on the same, I'll say, risk field, if that's the right term for that. So I think it will be a combination of those things I mentioned.
Thank you so much, and thank you, our audience. What an energetic group. We actually have time to answer one more question today before we close, but the presenter will be emailing answers to the other remaining questions. Our last question from our audience member before we close is, what do you think are the major biological differences in having earlier cancers in African Americans?
great question. I'll tell you from some of the studies I've done and from studies I've heard of, I personally don't necessarily think it's the race that's different. It's probably a combination of the things that we eat that shape the environment in the colon. And this comes down to probably the local microbiome and other metabolic parameters that are within the colon. So the way I always think of things, I started out with a presentation talking about taking a genetically susceptible individual and exposing them to the environment. And in the colon, the biggest driver of the environment are the things that you eat and then are metabolized. Can you imagine if, and so we know that, for instance, high-fat diets high in red meat, the so-called westernized diet does put anyone and everyone at a much higher risk for colorectal cancer. And we know this because some of the other studies in countries in which are not on westernized diets, there may be genetically susceptible individuals there, but they don't develop colorectal cancer. I always think of the Japanese as a great example. In pre-World War II Japan, we've had, who are largely on a rice and fish diet, their rates of colorectal cancer were pretty low compared to the United States. But immigrants who came from Japan to work in the United States pre-World War II on the west coast of the United States, within 1 or 2 generations, had rates similar to the United States. That tells you that this is largely not genetic for that difference, but that the environment, in this case, the type of food that they eat over those generations, probably influenced, maybe other things, tobacco use and other things. I also look at what happened in post-World War II Japan. Post-World War II Japan, after the World War II ended, there was a renaissance of building and rise of economic power. And many of the Japanese ate more westernized meals. There was still the coastal that ate the rice and fish diet, but those who went to a more westernized, there was evidence that their rates of colorectal cancer surpassed that of the United States because of their westernized diet. So in that case, I would say that the Japanese were probably always genetically more susceptible to colorectal cancer, but you never saw it because they never ate the westernized diet until that manifested itself at the right time in our history. And now their colorectal cancer rates are higher than the United States. I would say the same thing is probably in African Americans. There's probably certain diets, particularly many African Americans live in inner cities or poor areas, and this may be true for even non-African Americans in poor areas, where their microbiome makeup, the type of food they eat, puts them at a much higher risk. And the great experiment would be to take that same person and put them in a different environment, give them a different socioeconomic class, give them a different diet, and watch them over the next 10 years, watch that microbiome change, and see if that reduces their risk for cancer. So I think that's a great question, but I think there's a lot of studies trying to answer that. They're complex studies, but I think it's going to come down to that local environment around in the lining of the colon that's largely driven by the stuff that we eat that probably makes those differences. But that's a hypothesis.
Thank you so much, Dr. Carethers, and thank you for your informative presentation and your important research. I'd also like to take the time to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share the announcements with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye-bye.